Hello, and thank you for joining us today for the webinar titled Biospecimen Quality for Precision Medicine. I'm Shonali Paul, COO at CloudLIMS.com. I'm here to moderate this event. Dr. Jim Vaught, Editor-in-Chief of Biopreservation and Biobanking, is joining us today as the presenter. Before we get started, I'd like to go over a few points so that you know how to participate in today's event. All attendees are in mute mode throughout this webinar. This is an interactive webinar, so we encourage you to ask questions anytime during the presentation by entering them in the chat box and clicking send. The questions will be answered during the Q&A session at the end of today's presentation. We'll take as many questions as time permits. However, if we are unable to answer your questions today, we will answer them offline. It is time now to hand over the floor to Jim.
Good afternoon. I'm Jim Vaught. I'm Editor-in-Chief of Biopreservation and Biobanking, and I'm also a guest professor in Changsha, China. Today, on behalf of CloudLIMS, I'm going to be presenting a webinar about biospecimen quality for precision medicine. So let's get started. So here are the key talking points for today. What is a biobank? We'll try to define it. What is the role of biobanks in precision medicine? What are the requirements for research for precision medicine involving biobanking? What are some of the factors researchers should consider when sourcing biospecimens from biobanks? Why is biospecimen quality critical? What are the key elements of maintaining biospecimen quality? What are some of the approaches to improve biospecimen quality? What is the role of biobank informatics in biospecimen quality? So what are biobanks? Generally, we think of them as entities that receive, store, process, and/or disseminate specimens such as blood, tissue, urine, saliva, etc. as needed. And the biobank encompasses the physical location as well as the full range of operational activities. It can be one freezer or a freestanding entity, virtual or part of an institution. And we would like for Biobank to have a professional staff and commitment to maintain and preserve specimens and records for future reference and historical continuity. But having a professional staff often depends on the size of the biobank. The more larger commercial biobanks are more likely to have a professionally trained staff. And biobanks also may be called biorepositories, biospecimen resources, or biological resource centers. And you'll see these terms used in various ways in various publications, but biobanks is the most common term these days. Biobanking is multidisciplinary. There are many types of expertise that are involved in biobanking. We'll concentrate on human biospecimens and biobanks in this presentation, but human biobanking can involve any of the following: pathologists, epidemiologists, clinical chemists, also biochemistry expertise, as well as other clinical, basic, and translational research expertise. And other than human biobanks, there are many other types of biobanks and related collections, including cell culture, museum collections, seed banks, and also collections from sea life for drug development, for example. So what are the types of human biobanks? The European Biobanking and Biomolecular Resources Research Infrastructure, or BBMRI, discusses two major types of biobanks: population-based biobanks, where the general population are sampled to study certain types of disease processes, and also disease-oriented biobanks, where samples are collected from people with particular types of disease to study that disease, along with control samples so that you can tell the difference between the disease samples and control samples when they're analyzed to help you study the origin of certain disease types and various factors that may affect the development of certain diseases. So what do biobanks store? They store various types of samples, which we'll get into in the next slide. but also generally want to store for clinical studies the clinical history of patients, demographic information such as diet, smoking, alcohol consumption, that sort of thing, but also the family history, the history of certain disease types that you may be studying that a family may have been exposed to in the past. Also, for the samples, you want to know about their sample collection procedures, processing and storage, and store that kind of information in your informatics system. And also information about informed consent if the samples are collected from patients for studies that require informed consent, and also information about data safety and protecting the privacy of patients through, for example, the HIPAA regulations in the US. So what are the types of biospecimens that are stored? You may want to store whole blood. That's a very common sample to collect. And often the blood samples are divided into plasma, buffy coat, serum, and also may be a source of blood DNA and RNA, stem cells, white blood cells, and those sorts of derivatives from whole blood. And in addition, biobanks often store urine, various types of tissue, maybe disease tissue, tumor tissue, or control tissue from all parts of the body, and also cerebrospinal fluid, synovial fluid, and also not shown here, but one of the major samples stored in biobanks these days is saliva, which is a good source of nucleic acids and also can be used for proteomic studies and other biomarker studies. So what is precision medicine? The term precision medicine is often described as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. So previously in medicine, there was a one-size-fits-all in terms of diagnosis and treatment, and all patients were pretty much treated the same way. And then stratified medicine was the next big step, where patients were grouped by disease subtypes, demographics, clinical features, and biomarkers. And now we've moved into the era of precision or personalized medicine, where patient individual preferences, clinical features, medication history, environmental exposures, behaviors and habits, and biomarkers are used to treat patients individually according to all of those factors and others that physicians may take into account. So what is the Precision Medicine Initiative? This initiative is now called All of Us in the United States, and it was announced by President Obama in his 2015 State of the Union address. And so its mission is to enable a new era of medicine through research, technology, and policies that empower patients, researchers, and providers to work together towards the development of individualized care. Before the initiative was started 3 years ago, Drs. Collins and Varmus wrote a perspective piece in the New England Journal of Medicine that described the various benefits of precision medicine, the various types of medicine and disease processes, and concluded that precision medicine would lead to a better understanding of underlying mechanisms by which various diseases occur, improving diagnosis and treatment, and could result in some newly classified diseases. So what are some of the challenges of precision medicine? The data collected by researchers in clinics and hospitals, for example, need to be standardized. There needs to be standardization of the ethical, legal, and social issues, also abbreviated as ELSI, which include privacy and the confidentiality of donor health information and the informed consent documentation. So there is a huge business aspect or cost to precision medicine. The initiative itself will cost many millions of dollars. It involves the development and advancement of new technologies. DNA sequencing is currently very expensive to carry out, but the sequencing cost is decreasing quickly. And also drugs that are developed to target a person's genetic or molecular characteristics are likely to be expensive. And the cost of collecting, processing, and storing samples for the initiative is going to be very expensive as well as the analyses that lead to the improvements in precision medicine. So the role of biobanks in precision medicine, as we've said, it's an organization that acquires and stores human blood and tissue and the associated genetic data for future research use. And they provide an opportunity for more efficient research by providing access to large amounts of genetic and electronic health record data. So the cost of precision medicine will decrease over time, but will have a large initial investment in biobanking and sample collection. So we need to establish strong biobank practices that could facilitate research that will enable us to ultimately inform primary care and allow doctors to create the best individualized treatment for their patients. This slide shows the role of biobanks in precision medicine in a graphical way from the upper right-hand corner, collecting the sample from patients, processing and storing the sample in a biobank in the bottom of the slide, the analysis of biospecimens, molecular data, proteomic data, etc., that is part of the translational research process, and leading back to the upper right to the diagnosis and therapy of patients through personalized medicine. So the biobanking requirements for precision medicine are at least threefold in this slide. High-quality biospecimens are obtained from carefully characterized individuals with detailed medical records. The specimens and anonymized data are collected, and biospecimens need to be available to researchers who can show a particular need for the samples and data, and there will have to be a committee, an access committee in the biobanks that will study the proposal of various researchers to use the precision medicine samples and data and approve or disapprove the use based on the quality of the application. So what are some of the factors to consider when storing biospecimens? Regulatory compliance is one general category. HIPAA, the privacy regulation and informed consent, and also the institutional review board approval of the study through the informed consent and other factors that may allow the IRB to approve or sometimes disapprove a study. The control, sourcing, and storage of samples through the appropriate treatment of biospecimens from collection and preparation or processing to transport, and of course the storage conditions. So some of the factors that researchers should consider when sourcing biospecimens from biobanks are that they need to be well characterized and properly annotated with data, and there needs to be proper adherence to the requirements of 21 Code of Federal Regulations in the U.S. about specimen management systems and data. And then there needs to be proper data and specimen management systems, data security, so that improper sharing of data is not done and privacy is protected, and then at least a daily backup with redundancy at an off-site secure data center. Why is biospecimen quality important? Because the sample quality affects the laboratory test results, as we'll show in the next few slides. Samples require sufficient phenotypical data, control of the chain of custody, and access to and control of any constraints in any informed consent documents. A few years ago, I worked with some colleagues on a clinical chemistry article called the Q&A, where experts from Africa, the UK, Canada, and the US discussed these various critical issues in international biobanking. And on this next slide, it shows the major conclusions from that clinical chemistry article, and that the current critical issues are related to quality management and controlling specimen pre-analytical variation, which we'll talk about in the next few slides, centralized versus distributed or local biobanks, long-term sustainability of biobanks, which gets into the business planning of biobanks, which is a huge issue these days that we won't talk about unless any of you have questions after the presentation. Communicating the potential value of biobanks to stakeholders, returning research results and incidental findings to donors or patients, which is also a controversial issue that we'll not talk about, but I'll be happy to answer questions if you have questions about that aspect of biobanking. and also data management and sharing. So maintaining biospecimen quality involves various factors. We're going to be talking about pre-analytical factors that affect the quality of samples in collecting, processing, and storing samples, and then analytic factors that affect the performance of a particular testing procedure in a laboratory. So I wrote a review article with a colleague a couple of years ago in clinical chemistry that discussed the various pre-analytical phases, analytical phase, post-analytical phase, and so forth with regard to clinical chemistry analysis of samples. You can see that in terms of biological and environmental factors, anything from the time of day or season of the season of the year that the samples are collected, to coffee consumption, alcohol consumption, weight, smoking, medicines a patient are taking, and even exposure to sunlight can affect the quality and the results of analyses of samples. And also there are other technical factors in the collecting, processing, transport, and storage of samples that may at any of these stages affect the quality of the analyses of samples. And also in the analytical phase, the instruments that are used, making sure that the instruments are validated and they're giving accurate results, the quality of the work by a technician that's analyzing the samples may also affect the quality of samples. And then after the samples are analyzed, the quality of the way the results are analyzed, the statistics applied, which may not be proper for that particular type of analysis, may also affect the outcome of a study. So some of the key elements and pre-analytical factors are the physiology of the human research participant prior to collection, as we discussed in the previous slide, the uniformity of the collection practices through standard operating procedures, planning for the proper annotation of the samples prior to collection and removal of samples. This is very important. You may only have one chance to decide on the types of data elements that you want to collect for samples, and those need to be discussed in detail before any samples are collected. Optimizing the biospecimen handling practices prior to their inclusion in downstream testing. Some of the analytic factors are the use of validated assays. This is very important. Use of standard procedures where the technical staff is well trained. The uniformity of reagents. The lot uniformity is important so that all samples collected with the same reagents give proper, accurate results. The inclusion of appropriate type and number of quality control or reference samples. Randomizing the sample population or the patients or donors where possible so that analyses are more uniformly done and accurate. And standardized methods for documenting and interpreting testing results. Some other factors are collecting the samples based on the mission and goals of a biospecimen resource, handling of biospecimen storage according to SOPs specific to the biospecimen type. Various types of samples need to be stored in various ways. For example, cells that may need to be kept viable over a long period of time usually will need to be stored in liquid nitrogen or liquid nitrogen vapor as opposed to refrigerated or other types of freezers, minus 20 degrees or minus 80 degrees, that may be proper for other types of samples that are not so sensitive to temperature. Use of biospecimen resource SOPs to maintain sample quality, and also consideration of shipping conditions that may need to be different for different types of samples. Again, to maintain the viability of cells, you may need to ship in liquid nitrogen vapor containers. When I worked at the National Cancer Institute, we developed best practices for biospecimen resources. We tried to harmonize biobanking practices across the National Cancer Institute and NIH enterprise. We worked with various committees. We performed various studies that helped us fine-tune our best practices, as shown in the fourth bullet point here. We recommended partnering with accreditation and professional societies such as the College of American Pathologists that has a biobanking accreditation program to ensure that implementation and integration of such practices made it into the biobanking enterprise and medical enterprise in general. And then we had a biospecimen research program at the NCI that you can find documented on the NCI Biospecimens and Biospecimen Research branch website that I'll give you at the end of the webinar if you'd like to look further into the details of these programs. So what is the role of informatics in biospecimen quality? Biospecimens need to be tracked from the time that they're collected, processed, stored, and shipped out to analytical laboratories, and that there needs to be a comprehensive IT program for tracking. But also you need to collect data in an informatics or LIMS system about the clinical information from the patient, the quality testing of samples, demographic information about the patients. You need to identify, as I said before, the data elements that are necessary for each study, and that needs to be done before you collect the first sample to make sure you're collecting all the information that you want to collect for your particular study needs. There has to be data security and protection of privacy of patients, and especially for multi-center studies, it's very important to have systems, informatics systems that are interoperable, that can talk to each other, so to speak, so that you have a comprehensive view of everything that's going on at every center, and the data can be processed and analyzed across all of the centers and the data processing center equally. So this webinar is sponsored by CloudLIMS, and their mission is to promote cloud-based technologies for biobanking. And this graphic shows some of the advantages of cloud-based systems, such as low cost, also scalability. It can be expanded or contracted as needed to store all the information you need in the cloud for your study. And also there's low maintenance and low need for IT personnel. So these are some of the advantages of cloud-based informatics that you might want to ask the cloud-based informatics experts at CloudLIMS about. And on the behalf of myself and CloudLIMS, I want to thank you for listening, and we'll be happy to answer questions. Thank you.
Thank you, Jim, for the insightful talk. We have a number of interesting questions, so I'll move straight into those. So the first question is, do you recommend performing QC on tissue samples, or are there any specific guidelines for tissue samples?
Well, yes, in general, the quality control assessment of any samples, blood, urine, liquid samples, and tissue samples is advisable. Sometimes there's a limited quantity of tissue samples, and also the type of QC you perform, of course, depends on the type of study you're doing. When we were doing the Cancer Genome Atlas at NCI, we had both pathology quality control performed as well as quality control on the nucleic acids that were extracted. And so that was a way of making sure that the samples passed QC and entered into the genomics and genomic studies that were done by TCGA. So anyway, it's very dependent on the type of sample and the type of.
Another question we received is QC performed on frozen biofluids such as urine or blood samples?
Yes. Well, presumably the QC is performed when they're thawed, but of course there are, I know many of the attendees on the call will be familiar with the cryo extract where you can take samples. Otherwise, it depends on the type of QC you're doing. When I worked in the NCI epidemiology program, we would insert about 10% replicate quality control samples throughout the batches of samples that were being analyzed, and then make sure there were consistent results among all the quality control samples. There was no drift in the assay results. And again, it depends on the type of analyses you're doing, but certainly any kind of program, epidemiology or other type of research requires some sort of quality control. And for any of these questions, I can provide publications or slides if anybody's interested.
There is also a question about the QC best practices. I know that this is a long topic, but maybe you can touch upon the key best practices.
Key best practices in quality control or quality management? Is that the
Yes,
question?
on quality
Okay,
control.
sure. And again, I can provide some publications about this if anybody's interested. But the best practices documents usually will recommend overall a quality management program, which includes quality assurance, meaning that you have documentation of how your quality management program should be operated. And then quality control is the actual testing procedures that a laboratory or biobank need to put into practice, such as, well, for example, for freezers, regular temperature monitoring that can be documented. And then in the laboratory and biobank, a set of standard operating procedures that covers all the operations of the biobank and laboratory if it's associated with the biobank, and that technical personnel are trained in those standard operating procedures and tested periodically to make sure that they're aware of how the standard operating procedures work. And then there are ways to, after a QC assessment is done periodically, maybe every 3, 6, 9 months in a biobank to check the freezer records and so forth. Then if there are deficiencies that need to be reported to your quality manager, quality management leadership, then you can do that. You can file deficiency reports, and you can also write reports about how you're going to fix those problems that showed up in the quality control. That's sort of a general statement. If you're dealing with the FDA, then those are very serious issues, identifying and correcting deficiencies, which may result in your program having to take a break from processing samples until you've corrected all of your QC issues. But that's the general thing, having SOPs, having a organized quality assurance, quality control program, and quality control personnel who check for compliance with SOPs periodically.
Then there is a question on precision medicine itself. That should it move forward, and would you recommend preserving DNA for patients if minimum tissue is accessible for future research?
Collecting DNA if
So
tissue
the
is
second question is not very clear. Yes.
okay. Let me answer the precision medicine one first. That should the program go forward? Was that the general question?
Yes.
Okay. Well, the Precision Medicine Initiative in the U.S., which is now called All of Us, as I mentioned on one of my slides, is going forward. There was a large contract awarded to the Mayo Clinic in Minnesota, and they're the central sample collection processing biobank, and they expect to have 1 million contributors to that across the U.S. population, which is going to result in probably 10 to 20 million samples in the biobank. And they have a program laid out to do a lot of various testing procedures, analyses, including single cell analyses. And all of this is outlined in the allofus.nih.gov website, or if you just put in all of us into a search, then it'll pop up as one of the top 2 or 3 choices. So you can look on the website and see what they're doing. Should it continue as both a a scientific and a political question, but as far as I know, it's been funded for a number of years under this contract to the Mayo Clinic, and so it is going forward. And I'm not in a position to say how long it should be funded or whether it should be a long-term proposition. But so far, it's going forward.
And is there a shelf life for tissue samples stored in liquid nitrogen phase?
Um, Actually, um, long-term storage in liquid nitrogen vapor or in the liquid phase is the best way to maintain cellular and tissue viability. And as long as there are no thawing and refreezing incidents in the freezers, and they should last for many years. Um, Actually, the best expert on this kind of question is Alison Hubble, who is at the University of Minnesota, and she's written a couple of nice reviews in biopreservation and biobanking that I'm happy to share that goes into the stability of many types of samples and many types of analytes. There are a couple of huge tables in her review from 2 or 3 years ago that go into a lot of this. So if anybody's interested, there are some specific guidelines on what kinds of tissues and what kinds of analytes survive long-term and under various storage conditions.
Then there is a question on pre-analytical variability in biosamples. So if while collecting samples, some variations happen in the normalization of factors, how can it be managed in quality assurance?
Sure. Well, that's a very big question, and pre-analytical variables. And as I alluded to in the presentation, I worked on a couple of review articles about this issue, and there's a lot of work to be done. As I mentioned near the end, the NCI Biorepository and Biospecimen Research Branch had a biospecimen research network a few years ago that awarded grants to study some of these pre-analytical variable questions. And also there is on the NCI BBRB website, there is a biospecimen research database that has, I think at this point, over 2,000 publications. And the mission of the biospecimen research database is to compile publications that address these pre-analytical variable issues, and you can enter a type of analysis, a type of sample, etc., into a search engine, and the papers addressing that issue will pop up. The problem with pre-analytical variable studies is that there's so much conflicting data. So it's hard to say for any given tissue and any given analysis what all of the pre-analytical variables are that may affect the quality of the sample for analyses. But there have been some papers that show that collecting samples and stabilizing them either by freezing or in formalin or fixing in formalin quickly is the best way to avoid pre-analytical variables. And again, there's a lot of conflicting data, but when in doubt, it's best to try to stabilize samples as quickly as possible. And I'm sorry for the train going by my house right now, but if anybody hears that.
No, we didn't.
Okay. Okay.
Unfortunately, we are running out of time now and are unable to take further questions. However, if there are any further questions, we are happy to answer them offline. I would like to thank each one of you for joining us today. Have a great day ahead. Thank you.
Thanks, Ernal.